Hello! Good day, everyone! Welcome to Parasitology Part 3. In this lesson, we will discuss more groups of parasites. The parasitic fungi. Some estimates suggest that there may be over 1 million species of fungi, although less than 10% of these have so far been described. Unlike plants, fungi are heterotrophs, that is, they cannot make their own food and have to gain their nutrients by breaking down existing organic matter. The majority of fungi gain these nutrients by acting as saprophytes, that is, they break down dead organic matter. In addition, many species are in symbiotic relationships with plants and invertebrates, while some are parasites of other fungi, plants, and invertebrate and vertebrate animals. Some of these parasitic species are important in human and veterinary medicine, as well as wildlife ecology. For example, pneumocystis is a major cause of morbidity and mortality in AIDS patients. The skin disease ringworm in cattle is caused not by a helminth but fungi such as Trichophyton verrucosum and Clytridiomycete fungi are responsible for widespread and catastrophic levels of mortality among amphibians in many parts of the world. However, only the Microsporidia will be covered here. One member of kingdom fungi is Microsporidia. The Microsporidia are a cosmopolitan group of obligate intracellular parasites that infect a wide variety of invertebrate and vertebrate animals. Over 1,200 species have been described, but the majority of these are parasites of invertebrates and fish. Several species are of medical, veterinary, and commercial importance. For example, Nosema apis and Nosema sarini are major causes of disease in honeybees, while several species such as Nosema locustae and Nosema aegiri have been investigated for their potential as biological control agents. The figure shows the life cycle of Microsporidia. The spore is the only Microsporidian life cycle stage capable of surviving in the external environment. Immediately above the spore's plasma membrane are two protective layers. The first of this is the endospore, this one, which contains chitin and is electron-lucent when viewed with a transmission electron microscope. And above this is the exospore, this one. This contains a glycoprotein and is electron-dense, so it appears dark in transmission electron micrographs. The spore walls must provide excellent protection, as in some species, they can remain infective for over a year. Microsporidia are normally transmitted horizontally as a result of the host coming into contact with the infective spores. Humans usually become infected by ingesting or breathing them in. Vertical transmission has been described in those species infecting other animals as a result of crossing the placenta or the transplacental transmission in mammals, or infecting the eggs while they are still in the ovary in invertebrates. Transovarial transmission is common among endosymbiotic bacteria, which is presented in Wolbachia, but very rare among protozoan parasites. Once within the host cell cytoplasm, the sporoplasm, this one, the sporoplasm differentiates into a stage called a meronth and undergoes a series of asexual reproduction, which we call as merogony, in which numerous meronths are produced. Then after a period of merogony, the parasites start to produce spores. By the way, in this section, there is production of multiple meronths through binary fission. Then this meron transforms into a sporont, which produce the sporoblast. Then these sporoblasts will mature into spores. The spores steadily accumulate in the host cell and may eventually fill it. When the host cell membrane eventually ruptures, the spores are released and may infect the adjacent cell or be released into the environment. Another group of parasites are the parasitic plants. Although plants are normally considered to be primary producers that provide food for their needs by photosynthesis, there are a number of species that supplement this either partially or entirely by parasitizing other plants. In extreme cases, some of these species no longer produce chlorophyll. For example, the Indian pipe or corpse plant, Monotropa uniflora, which obtains its energy from mycorrhizal fungi that are themselves in a symbiotic relationship with tree roots. Plants do not, however, parasite animals, although some, such as the Venus flytrap, Dionaea muscipula, are carnivorous and obtain some of their nutrients by capturing invertebrates and digesting them. We also have another group of parasites. These are the helminth parasites. Invertebrates are multicellular animals that are united simply in their lack of backbone rather than the presence of any commonly shared morphological features. Over the years, various invertebrate phyla have been recognized and grouped together in many different arrangements. Molecular phylogeny can confirm some of these arrangements while questioning others. Consequently, readers should not be surprised by the lack of consistency between the texts. For the purpose of this lesson, we will be following the arrangement suggested by Brska, that is based on morphology and embryology. So under this scheme, animals are initially placed into one of two groups, the Parazoa and the Eumetazoa. And Parazoa are organisms that lack true tissues, and this group contains only the phylum Porifera, better known as the sponges. All sponges are aquatic and they gain their nutrition through filter feeding and, in some cases, symbiosis with algae or bacteria. Sponges, which belong to the family Cleonidae, bore into the shells of mollusks and penetrate the calcareous skeleton of corals, and they have been held responsible for causing mass mortalities in commercial oyster beds. Other sponges will, in their turn, exploit the burrows and also grow over them. Sometimes this is referred to as an example of parasitism, although this is debatable. The Eumetazoa are those animals in which muscles, nerves, and other tissues are recognizable, and this group contains the vast majority of animal phyla, all vertebrates and most invertebrates. The Eumetazoans can be divided into two subgroups, the diploblast and the triploblast, on the basis of their embryonic development. And the helminth parasites will the members of the phylum Platyhelminthes are commonly known as the flatworms, on the not reasonable basis that they are dorsoventrally flattened and worm-like in appearance. They are acelomate, soft-bodied animals that are bilaterally symmetrical, with an obvious head and this one. Although in some free-living species, the mouth is situated at or close to the center of the body, while in tapeworms, the mouth is absent. Tapeworms also lack a gut, although this is present in most other platyhelminth species and usually consists of a blind ending sac or a series of branching tubules. The lack of the anus means that waste material is ingested through the mouth. Platyhelminths too, however, have a ramifying series of tubules that constitute their excretory system. Flame cells, or the protonephridia, maintain the movement of fluid within the excretory system, and the waste is removed through excretory pores. Because the excretory system is used to remove excess water and ions, it is sometimes referred to as the osmoregulatory system. Most platyhelminth species are hermaphrodites, containing both male and female reproductive organs, although in a few species there are separate male and female sexes. There are four classes of platyhelminths. We have Turbellaria, Monogenia, Trematoda, and Cestoda. The parasites belonging to the class TREMATODA are commonly known as the flukes. Those living in the blood are therefore called blood flukes. Those living in the gut are intestinal flukes, while those associated with the liver and gallbladder are called liver flukes. The term fluke has been used in common language to describe the parasite we now know as Fasciola hepatica for hundreds of years. The class Trematoda is divided into two subclasses, the Aspidobothria and the Degania. The Degania has two clades, the Diplostomatida and Plagiorchida. The Diplostomatida contains the blood flukes, of which the Cystosomatidae or cystosomes are of importance as the cause of schistosomiasis in humans. The Plagiorchida contains the majority of the Guinean species, of which the families FASCIOLIDAE or Fasciola hepatica, Dicroceolidae, and Paragonomidae are of particular importance in human and veterinary medicine. This family, Fasciolidae, includes some of the largest flukes, Fasciola gigantica, which can reach 75 millimeters in length and 12 millimeters in width, while Fasciola magna is even bigger and can be up to 100 millimeters in length and 26 millimeters in width. Adult fasciolids are usually found in the liver and bile ducts of ungulates and other herbivorous animals, although omnivores such as pigs and humans are sometimes used as definitive hosts. So in this figure, it shows the adult Fasciola hepatica and Fasciola gigantica. This Fasciolidae or member of Fasciolidae is a Fasciola gigantica from African ox, and this is from Burmese ox. Letter C is from a British ox, and a Fasciola hepatica from British sheep. All parasites are to the same scale. Also, this figure is a scanning electron micrograph of an adult Fasciola hepatica from sheep. So take note that the oral sucker and the anterior of the body and the large ventral sucker. The genital pore is immediately above the ventral sucker. Take note that the dense array of spines which coats the surface of the worm. This figure shows the life cycle of Fasciola hepatica. Letter A is an adult form or the adult stage of Fasciola hepatica. So this releases the egg. This egg will transform to a miracidium, which will then become the sporocyst. This sporocyst will develop, becoming into a rhidia. The rhidia will transform into a tailed-like uh, form, which we now call as the cercaria, and this will become metacercaria. The young fluke will migrate to the liver. So the liver of the sheep will be infected once the metacercaria will be ingested through the mouth. Some cases it may be found in rabbits and in humans. The nematodes are simultaneously one of the most abundant, important, and yet neglected groups of metazoan animals. Some estimates suggest that there may be over 100 million species, but fewer than 30,000 have actually been described. Nematodes occupy all terrestrial and aquatic ecosystems, while one species, Halicephalus mephisto, lives over a kilometer below ground in a gold mine in South Africa. In marine sediments, up to 95% of the organisms present can be nematodes, and they are responsible for much of the benthic invertebrate biomass. Many nematodes feed on bacteria, fungi, and/or detritivores, but there are also examples of predators, and many are parasites of plants, invertebrates, and vertebrates. Most nematodes, especially the free-living species, are microscopic in size, but some of those parasitic in mammals can be several centimeters in length, while some giants are over a meter long. For example, the females of Pocentonema gigantissima, which lives in the placenta of sperm whales, can reach at least 8.4 meters in length and 2.5 centimeters in width. Adult Dracunculus medinensis emerging from the scrotum is shown in the picture. This species is a member of the family Dracunculidae, which causes the dracunculiasis. This is an initial site for the adult worm. Usually they emerge in the lower leg. In terms of abundance, numbers of species, total biomass, and exploitation of ecosystems, the arthropods represent the most successful metazoan animals. Over a million species of arthropod have been taxonomically described, and there are probably several times as many awaiting the attentions of biologists willing to study them. Some estimates suggest that there are currently about 1 times 1,018 individual arthropods living at any one time, and there is a possibility that some of them will be living on us. As a group, modern-day arthropods are generally small creatures with only a few marine species, such as the giant spider crab, reaching any appreciable size. However, 460 to 248 million years ago, they included the Eurypterid water scorpion, which were among the largest creatures alive at the time and grew up to 2.5 meters in length. So the arthropod. In the phylum Chelacerata, one of its members is the family Sarcoptidae. Well, this family contains the well-known scabies mite, Sarcoptes scabae, Nemedocoptes mutans, which causes scaly leg in hens, and a variety of other birds, and Notodrescati that afflicts domestic and wild cats, as well as dogs and a number of other mammals. So this is a female dorsal view of Sarcoptes scabae, and the male ventral view of the species. Unlike Demodix, in which there are numerous host-specific species, Sarcoptes scabi exists as a variety of host-specific subspecies. So, for example, humans are infected by Sarcoptes scabi var hominis, while dogs are infected with Sarcoptes scabi var canis. The subphylum Hexapoda contains the two classes, the class Incognita and the class Insecta. The class Incognita contains several orders of six-legged wingless organisms, which may or may not be true insects and whose common feature is that they do not have insufficient of the traits needed to be included in the class Insecta. The Pteroptera is divided into two suborders, the Maleophaga or biting lice and the Anoplura or sucking lice. They are all ectoparasitic on birds and mammals, and they are relatively host-specific, with most louse species feeding on a single host species or group of closely related host species. The Maleophaga have triangular-shaped heads that are broader than their thorax and biting mouth parts. They are predominantly ectoparasites of birds, although there are few species that feed on mammals. Most of them use their mandibles to chew on feathers, fur, or dander, but some of them will also consume epidermal secretions or gnaw through skin or quills to obtain blood. By contrast, the head capsule of the Anoplura is now narrower than their thorax, and they have sucking mouth parts that they use to penetrate the skin and consume blood. The mouth parts of the Anoplura are unlike those of most other blood-sucking insects. The labrum is modified to form a snout-like process called the haustellium, and when the louse feeds, the haustellium is everted. This brings into play a series of chitinous teeth that are now on the outer surface and anchor the haustellium in the host skin. The louse then employs three stylets to form two channels. Down of this saliva is secreted and up the other blood is sucked. The Anoplura have a more restricted host than the Maleophaga and only parasitize certain placental mammals. The Anoplura includes numerous examples of species of medical and veterinary importance. The two best known examples are, of course, the human head louse Pediculus humanus capitis and the human body louse Pediculus humanus humanus. Both species are known as common cosmopolitan parasites of humans. The body louse is thought to have evolved from the head louse at about the time humans started to wear clothing. The body louse has the smallest insect genome sequence to date. That this encodes for 10,773 protein coding genes and 57 microRNAs. Its small size is predominantly through losing genes involved in environmental sensing and those that code for detoxification enzymes. This is to be expected in a parasite that lives permanently upon its host and relies on it to provide food, shelter from the environment, and dispersal. Like other animals that live entirely on blood, body lice and head lice are reliant on a symbiotic relationship with microorganisms to provide certain essential nutrients. In the case of head lice and body lice, they contain an endosymbiotic bacterium, Rhizia pediculicula, within a mycetome. This bacterium provides the lice with pantothenic acid or the vitamin B5. Rhizia pediculicula also has an unusually small genome that encodes less than 600 genes. These do not include antibiotic resistance genes, and it may therefore prove possible to develop new means of controlling lice by targeting their symbiont. Order SIPHONAPTERA, or commonly known as fleas. The fleas are an unusual group of insects whose Relationship with the other insect order is still uncertain. They are all rather small, 1 to 10 millimeters, wingless insects that are parasitic on birds and mammals during their adult stage. The human flea, Pulex irritans, is becoming uncommon in industrialized countries, and in the UK and Europe, its place is being taken by the cat flea, the Tenosiphalides fellis. Pulex irritans uh, remains common in many developing countries and is a potentially important vector of diseases. For example, in a survey of 12 villages in Tanzania, Lodisa uh, it was found that Apulix irritans in 72.4% of houses, and unlike the other flea species that were present, its density was positively correlated with higher frequency of bubonic plague. Although it is called the human flea, Pulex irritans is also commonly found on sheep, cattle, goats, pigs, and even chickens. The dog flea, Tenosiphalides canis, has also been replaced by Tenosiphalides fellis in many regions, so the most common flea found in dogs is now usually the cat flea. That is all for the part 3 of our parasitology lesson. I hope you learned something. Thank you and have a great day. Bye.